Okay, our topic is the process of science, and your target is I can identify and conduct a controlled experiment. Okay? Now, yeah, you may be thinking some of these things, like maybe there's some confusion there. The steps, what is it? Or you maybe have memorized steps before, but you can't actually do it. Okay, so we'll try to answer those as we go through the notes today. So I wrote down a couple definitions or from various online dictionary sources, and they do get a little confusing. These last two are a little more simple. This one for sure. My definition would be to test one thing. Okay, so when you do an experiment, you're testing to see if one thing worked. Okay, so these are also, I believe, listed in your notebook. Okay, we start with a question or a problem or some kind of an observation or something you're interested in or confused about. That's where we start. For example, do plants grow taller when watered with flavored water? Or is bottled water better than tap water? Or do dogs see colors? Or why do high school students smell so bad? Whoa. After you decide on the question, that the problem that you're having, the thing that you want to answer, then you just do some research and you get some background information on it. And after you get the background information, then you need to make a hypothesis. And a hypothesis is a testable statement. It is what you think is going to happen based on the research that you did. Some examples of hypotheses are here. The last one down here is the one we're going to kind of use as we go through the video, and that is, if fertilizer is used, then plants will grow taller. That is a testable statement about plants, and so that's going to be our example. Okay, so after you make the hypothesis, you got to test it. Now, how do we test it? Okay, we set up and conduct a controlled experiment. That's what a controlled experiment does, is it tests a hypothesis. And if you're confused about what that is, a controlled experiment is where we're testing one thing. Okay, everything else is the same except for one thing, and that's what we're testing. Okay, so this is a term in your notebook. It's the independent variable, and this is part of a controlled experiment. Okay, and an independent variable is the thing you are testing. Okay, so if we're using this hypothesis here, if fertilizer is used, then plants will grow taller. The independent variable is the thing that we're testing. So based on that hypothesis, what is it that you think we're testing? I'll give you a couple hints. Still don't have it? Okay, there it is. So our independent variable is fertilizer, because that's what we're testing. We're testing to see if fertilizer helps plants grow. Okay? A dependent variable, that is the thing that you measure. So our hypothesis is going to be the same. If fertilizer is used, then plants will grow taller. What are we going to measure? What's our dependent variable? I got a couple other hints for you. Got it? Growth. Okay, that's what we're going to measure, whether the plants grow taller. So those are the two variables. The independent is the thing that you're testing, and the dependent variable is the thing that you measure. Okay? Now, as we're setting up our controlled experiment, we've had our question, we've done our research, we've formed our hypothesis, okay? And we got to create a couple groups, okay? So we're going to, at least two groups, and over here, one group gets the experiment, or one group gets the independent variable, and the other one doesn't. This little flask here is nitrogen, that's fertilizer. So our experimental group is getting the fertilizer, or the independent variable, okay? The control group is not getting the fertilizer. So then the question is, why do we need that second group? Okay, what do we need a control group for? Why don't we just add fertilizer to it and measure how tall the grass grows? Well, we have to have something to compare it to. Because if we don't have a control group, we don't know if it was the fertilizer, or if that's just how tall grass grows. Okay, so we need the control group for comparison. Okay, so we've got an experimental group that gets the independent variable and a control group that does not. Everything else is the same. Okay, so the only difference between our two groups in this example is the fertilizer. All these other things Sunlight, water, temp, soil, size of the pot, all those things have to be the same. The only difference is going to be fertilizer. Okay, when we get to recording our data, this is a data table. That's where we write down our basically our raw results that we get. We're just counting or tallying things up, and then these are different types of graphs that we could use. So when we record data, you need a data table and you need a graph, both of those things. Okay, once you get that done, then it's time to make your conclusions. Okay, so you state whether your hypothesis was correct or incorrect. Either way is fine. If it's correct, great. If it's incorrect, great, as long as we find out for sure. Okay, if you find out if it was correct or incorrect, that means that you set up and did your experiment the way it should be. We don't want it to be like, well, we're not sure. That must mean there was an error or you didn't set it up right. Okay, so we're stating our hypothesis. Was it correct or was it incorrect? And then we are summarizing our data. Now, what that means is that the data should be an explanation of your experiment and your results, but you're not going to be referring to your graph or data table. You want to write that as if you were talking to someone over the phone. Okay, so just they're not going to have access to your graph or data table, so it has to be explained in that way. Okay, so now what again? The steps or the general ideas that we started with a question or a problem. We did our research. We made our hypothesis. We set up a controlled experiment and tested the hypothesis. We recorded our results and we summarized our conclusions. That's basically it. Thanks for watching.